Well, here we are again. We are finishing up chapter 2, which is all about the chemistry of life and biomolecules. And we started in our last section to talk about the biomolecules, and there are 4 kinds: carbohydrates, lipids, proteins, and nucleic acids. So we're just going to pick up those last 2 categories of biomolecules. I want to remind you, to be organic, they have to have carbon, hydrogen, and oxygen in them. And how those are put together and what additional other elements are put in with them is what dictates if it's a carbohydrate, a lipid, a protein, or a nucleic acid. So now we're in section 2.6, and we're going to hone in on the proteins. The monomer subunit of a protein is an amino acid, and there are about 20 different amino acids, and the DNA dictates what order those amino acids are assembled. Proteins are important for diverse functions in the body, including your hormones, enzymes, antibodies, and even transport. The thing you have to be careful about with proteins, they're really important to how your body works. So things like enzymes that run chemical reactions, those proteins still need to be in a really precise shape. But they can denature, which is undergo a change in shape that causes them to stop working. So getting too hot will denature a protein. This is why if you leave a dog in the car and they get too hot, those proteins start to denature, which is why even when you cool them back down again, you can't recover the proteins and frequently will lose them. So what makes those different amino acids? Like I told you, there's 20 different kinds, and they all have a similar base structure. And if you look at all of these different amino acids, each one of them has a very similar base. However, you can see these little hats that are hanging on the end of them that are highlighted in gray. And these are the special groups that help make the different amino acids different. And that's why when we assemble those different amino acids together, we get different shapes of proteins. Proteins are organized into different levels. The primary organization is just the order the amino acids are placed in together. Secondaries, we take that long chain of amino acids and then they start to pleat or sheet up or form helices. Then that 3-dimensional shape starts to form, and it takes up an actual 3-dimensional functional group. And then we can even combine additional ones together. So we can take multiple proteins, each with their own 3-dimensional shape, and put them together into that quaternary structure. All proteins have a primary, secondary, and tertiary structure. Some will then be assembled to make that even larger molecule. But it's that 3-dimensional shape that allows proteins to function and do what they need to do. Here you can see a visual of it. There's that primary structure where you're just seeing the chain or the sequence of amino acids. That secondary structure, we start to coil up like a telephone cord, or you can see a pleat like folded paper. And then that tertiary structure where there's coils and pleats start to move together to form that 3-dimensional shape. And that quaternary structure where you can see multiple proteins have been then assembled together. Like I say, not everything makes it to that level, but we all get to this level, which gives us the functionality of the protein. The 4th category is our nucleic acids. And these have a monomer subunit of a nucleotide, which is formed from a sugar, a phosphate, and one of the 4 bases. So you can see a nucleotide here. We've got that one of those bases, then we've got our sugar, and then our phosphate group. This is where we have the coding for the order of the amino acids. Nucleic acids include our RNA and our DNA. DNA is where that code is stored. RNA is used in assembling the amino acids together. If you look at a nucleotide, we can see the phosphate group, we can see the sugar, and then there's a nitrogen-containing base. And there are 4 different kinds of, well, actually 5 different types of bases that can be added to this. The bases that are found in your nucleotides, we have A and G. These are double-ringed purines, and C, T, and U, which are single-ringed pyrimidines. In DNA, A pairs with T, G pairs with C. Hopefully that feels familiar to you. In RNA, the T is replaced with a U, but otherwise we have the same basic pattern followed. Here you could see an RNA molecule where you're seeing the single-sided strand, phosphate, sugar, phosphate, sugar, phosphate, sugar. Then we've got our bases that are hanging out on this edge. The order of those bases is dictated by the DNA molecule. Again, notice the U is used instead of T. Over here in the DNA molecule, you'll notice that it's a double-stranded molecule. We can see that phosphate sugar alternation again. And we see those bases. There's 4 different kinds, A, T, C, and G. C always bonding with G, A always bonding with T. And it's that order of the bases on the DNA strand that dictates the order in the RNA strand. So in summary, DNA has a sugar. The bases are A, T, C, and G, and it's double-stranded. RNA, the sugar is ribose, and the bases are A, U, C, and G, and it's a single-stranded molecule. So the macromolecules that we've discussed in this chapter are the 4 carbohydrate, lipid, proteins, and nucleic acids. We have the monomers of a monosaccharide and carbohydrates, fatty acids and lipids, amino acids and proteins, and nucleotides in nucleic acids. And then we can see carbohydrates are immediate, quick energy. Lipids are long-term energy storage. Proteins are support, metabolism, regulation, transport. They help us move like in our muscles. And then our nucleic acids are what store our genetic information. You guys have done a great job. Thank you so much.